I'm Ryan Weeks, and I want to talk to you today about how we use a fascinating biological tool to restructure the genome of a tiny fish to study a devastating human disease. The tool is CRISPR-Cas9, a bacterial defense mechanism that has been adapted to make precise cuts in double-stranded DNA. The fish is a zebrafish, and the disease is amyotrophic lateral sclerosis, or ALS, a debilitating and fatal disease that affects middle-aged and older adults. We have targeted a nuclear localized protein called TDP43 that in disease conditions irreversibly misfolds in clumps to form insoluble aggregates in the cytoplasm of motor neurons, a pathology that has become a hallmark of ALS. Previous work in another lab revealed that lysine acetylation is a post-translational modification of this protein that can invoke formation of insoluble aggregates. Follow-up work recapitulated this pathology in motor neuron-like cells by substituting the amino acid glutamine for lysine to create an acetylation mimic which produced similar ALS-like phenotypes. Here we describe in detail how we use CRISPR gene editing to make this point mutation in zebrafish to create a potential ALS-like model. Our goal was to generate a mutant fish that produces pathologies consistent with TDP43 aggregation that can be used to explore mechanisms of disease as well as advance our understanding of gene-environment interactions linked with neurological disorders. An in silico model demonstrates how a structural alteration could possibly change the function of this protein, as the lysine of interest is located in a domain responsible for RNA and DNA binding. We then explain how after making a targeted, double-stranded cut with CRISPR-Cas9, we bypass the default non-homologous end-joining pathway of DNA repair and used homology-directed repair to achieve the desired substitution, a repair that only has a 1 to 4% success rate in zebrafish. Our breeding strategy describes how we incorporated this mutation on an MNX GFP background in which motor neurons are GFP labeled, adding an additional layer of utility to our fish. Sanger sequencing confirmed the incorporation of this point mutation in our region of interest and the transfer of the mutation through the germline to successive generations. Characterization of this mutant fish began with light, dark, and sea startle behavior assays in zebrafish larvae. Subsequent Western blot analysis of arsenite-stressed aged adults following a swim tunnel experiment revealed increased insoluble TDP43 aggregation and protein ubiquitination, validating our model as a potential tool for studying proteinopathies associated with neurodegenerative disease. Ongoing characterization has generated additional intriguing questions about how this mutation affects the in vivo function of TDP43 that we plan to explore in the future. I welcome the opportunity to speak with you about details of this work and would greatly appreciate your feedback, so please visit me online or in person at the conference. And thank you very much for your time.